Can you describe your role as principal investigator in the trial and as chair of the clinical steering committee?
I'm honored to have that position as the head of the steering committee. I've been working in pulmonary hypertension my entire 30-year career. I've seen this disease evolve from a state where we didn't have a single therapeutic for this disease state, and we were transplanting all these young women, which is how I got involved in this, to now a disease state where we have therapies that can help people live longer, and that's important. Because now we have the opportunity, because they're making patients live longer, to really find newer drugs that remodel the blood vessels that actually can push the mortality of this disease way down so that these people can experience a normal length of life. And being able to share that totality of experience as a principal investigator for a new novel therapeutic, I think is very important. Because that brings a lot of experience to the table on how to design these trials and how to keep these trials on pace. Because I know the field very well, I've handpicked investigators for this trial that I know are top in their field, that appreciate the nuances of this combination of a technology with a new remodeling drug. And so one of my main jobs is to make sure that the trial moves on in pace, to encourage investigators, to keep them excited about the trial so that we get this information to design our phase 3.
Why did you choose to be so involved with Cereno in the CSI clinical trial program?
Well, I was very excited because Cereno is very forward-thinking and they appreciate innovation, and they were very accepting of using novel, evolving technologies and risk calculators in the construction of their phase 2. So when I see a company is very forward-thinking, I know they're really thinking about our patients. And that's the type of thing that really attracted me of working with Cereno, is there's a true appreciation of this disease state, and there is a true want to see this medication work appropriately to help our patients.
Tell us about the Cereno Scientific CS1 clinical trial.
The CS1 trial design is very innovative and very forward-thinking in that we are very deeply clinically phenotyping patients. We're using multiple modalities, including risk calculators, including the REVEAL calculator. We're using cardiac MRI to get a beautiful illustration of the function of the heart. We're also using echocardiography to do indirect measurements of pressures and pulmonary artery right ventricular coupling. And then we're using the CardioMEMS device, which is highly innovative, that will give us frequent measures of cardiac hemodynamics throughout the course of treatment so that we can determine the optimal dose of CS1 and also determine how often or how early the effects of CS1 come on and when their peak activity is.
How has the CS1 study been designed?
The trial design of this study is very innovative in that we are really making an effort to comprehensively clinically phenotype our patients. So we're doing multiple modalities to look at the structure and the function of the right heart, including MRI and echocardiography. The key thing here is that with MRI and with the CardioMEMS, which I'll talk about a little bit later, we can accurately depict right ventricular and pulmonary artery coupling, which is really the holy grail in determining prognosis in this disease. We're using multimodality risk factors to really give us a comprehensive idea of the risk of the patients that we're enrolling in the trial and the change in risk that this drug may impart to them throughout the trial. Using the CardioMEMS device, we will get frequent measurements of hemodynamics, which will tell us many things about the way CS1 works. It will tell us how quickly it works, when the peak activity is, and when the peak effect takes place, but it will also be able to tell us very nicely which one of the drug doses that we're applying in this study will be the best for a phase 3 trial. So we're garnering very important information, including even new biomarkers on thrombosis. So we'll really be able to accurately depict how this drug works and how well it works.
What makes this trial innovative?
I think the innovative aspects of this trial is that we're using risk calculators front and center. One, to enrich the trial with the right risk patients, and that's been a flaw in many of our clinical trials, in that we've been enrolling low-risk patients in these trials, and it takes a long time to reach an endpoint when you have low-risk patients who don't have endpoints. So we're using risk calculators to enrich this trial for the right risk patients, so intermediate risk, that likely will have events, so we could really test the efficacy of the drug. By using different imaging modalities like MRI or echocardiography, we'll have the most accurate depiction of right ventricular remodeling of any early phase trial that it's been done thus far. And marrying this imaging information with the ongoing and frequent assessments of hemodynamics using the CardioMEMS device, we can very accurately depict early signals of remodeling. And remember, that's the key thing about CS1, it's a remodeling drug. And so by able to depict early changes in hemodynamics, we really have an idea if we're doing something effective in remodeling the pulmonary arteries and the right heart's response to that improvement.
What are you hoping the results from this trial might show?
This phase 2 trial, I'm hoping that it's going to show that this drug is safe to use and that we will have key endpoint inclinations for our phase 3 trial. So by really phenotyping these patients well in this phase 2, we'll know exactly what to use in our phase 3 as a meaningful endpoint to really look at this drug. And then obviously we're testing several doses, so using the CardioMEMS device, we'll be able to accurately depict the right dose to use in our phase 3 trial. And obviously we're hoping for some hints of efficacy in this phase 2, that this drug does do exactly what we think it does, which is remodel the pulmonary arteries. And that's why having the CardioMEMS device that gives you so much data over time will really be very helpful in determining that.
What are the most prominent characteristics of CS1 that give you reason to believe in positive results?
Well, it attacks the disease from many different angles, which is things that we like in medicines, particularly in those that we actually expect to remodel tissue. It has antifibrotic activities, it has anti-remodeling activities, it has antithrombotic activities, and it also will help the right heart remodels. These are all things that we want in new therapies for pulmonary arterial hypertension.
What endpoints would be considered in a phase 3 trial to develop CS1 further?
So the phase 3 trial has to be designed with a very meaningful endpoint, and that's what we hope to determine by using a very well-characterized phase 2 trial. Given that we have so much information from this phase 2 on how this drug works, our phase 3 will be very nicely designed with features that we've learned from this phase 2. And so we'll be sure to have an appropriate endpoint that really will show if this drug remodels the pulmonary arteries the way we want it to and restructures the right heart in response to that positive remodeling that we hope to see with CS1.
Do you anticipate that diagnostic and monitoring devices, such as CardioMEMS, will be developed in conjunction with novel drug therapies?
The CardioMEMS device is such a useful device, and in these studies, like the CS1 trial and others that are using the CardioMEMS device in pulmonary hypertension, I think we're really going to find the utility of this very novel device that can alter the management of our patients in a very meaningful and early way. I mean, the earlier we can depict adverse remodeling or adverse cardiac response to that remodeling, the quicker we can act on it. And so it gives us a lot of lead time to have that information that we glean from the CardioMEMS device. And indeed, in these several trials that are utilizing the device, we will hopefully get a new device indication for the CardioMEMS for pulmonary arterial hypertension. So I do think that the use of this device will evolve with these trials.
Do you think that you're in an unusual space in that this is a marriage between technology and pharmaceutical?
It's great. This is one of the most exciting things about this trial is combining a novel hemodynamic device with a novel remodeling therapeutic And the information that you get by using this combination, I think, is really going to serve as a pivotal cornerstone for phase 2 trials in the future in this disease state that can use this same sort of combination.
For more episodes of our Insight series, please follow Cereno Scientific on LinkedIn or our YouTube channel.